Hello, everybody. I'm happy to share with you today our work on potential repurposed drugs against SARS-CoV-2. In this study, we studied the effect of 7 FDA-approved drugs currently under clinical investigation against 7 viral proteins related to viral replication and cell entry, in addition to 2 host proteins related to the cell entry. As we know, COVID-19 is the current pandemic covering all the world, caused by SARS-CoV-2 virus, and it has emerged in 2019 as a pneumonia outbreak in Wuhan, China. And until mid-February, there are more than 100 million cases and 2 million deaths worldwide. The viral genome, composed of single-stranded positive RNA of around 29 kilo base, which encodes about 10 genes, which encodes 10 genes. Some of them encode for polyproteins, not only single proteins, and these polyproteins undergo post-translational splitting later to encode for 24 to 27 viral proteins. Out of these proteins, 4 structural proteins form the viral capsid: the S, M, E, and N proteins. The first selected drug is the S protein, which is a spike glycoprotein responsible for the famous coronavirus shape. The S protein is proteolytically cleaved by type 2 transmembrane serine protease, TMBRIS2, into 2 subunits, S1 and S2. S1 binds to the host cell surface receptor, and S2 is responsible for the fusion of viral envelope and the cell membrane. The N protein is a nucleocapsid phosphoprotein, which binds to the viral genome and forms the viral nucleocapsid. The M protein is a membrane protein, which is one of the most abundant envelope proteins, and it determines the morphology of the virus, as well as being responsible for the interferon antagonist activity. Actually, the 3 structure of the M protein is not available in RCSP, so we had to model it using Swiss model server. Then the RNA-dependent RNA polymerase. This is a very important protein because the human host lacks this functionality. So there are lots of studies that target the RNA-dependent RNA polymerase. There are a lot of studies that target the RNA-dependent RNA polymerase being unique to the viral functionality. The main protease is a serine type protease. The other protease is the papain-like protease. And finally, we have the non-structural protein 14, which is essential for the RNA replication and transcription. The C structure of the NSB14 is not available as well, so we had to model it using Swiss model, using the C structure of the NSB14 of the SARS-CoV-1 virus. The 2 receptors of human origin are the ACE2 and the TMBRS2. The ACE2, as we know, is a human converting enzyme 2, and the virus uses it to enter the cell. And the TMBRS2 is a human transmembrane serine protease type 2, and the virus uses it for protein priming. The selected ligands we had used in our study are the famous antimalarial drug chloroquine with its derivative hydroxychloroquine, they are potent inhibitors of anti-malaria and show activity against some viruses. Ivermectin is a broad-spectrum antiparasitic, which show efficiency against some viral infections, in addition to RNA polymerase inhibitors, remdesivir and favipiravir. Remdesivir is a nucleoside analog originally developed for Ebola virus, and favipiravir, also known as Avigan, was developed in Japan for influenza, and it's effective against the RNA-dependent RNA polymerase. Our methodology in the study included docking of the ligands against the receptors that was performed using MVD version 6. Then molecular dynamic simulations of selected receptor ligand complexes that show good potential using GROMACS, followed by calculating the binding energy using MMBBSA GMMBSA package, employing the solvent accessible volume as a model for calculation of nonpolar solvation energy. Now I will talk about the results. In brief, I will talk only about the ligands which were found to be of potential importance. Ligands were found to be of potential multidisciplinary importance. They are ivermectin and remdesivir. Ivermectin binds well to the S protein as well as the human S2 and the TM BRIS2. Binding to both of them and the S protein indicates that it has a role in inhibiting viral entry into the cell, probably by increasing the endosomal pH. It also binds to both proteases as well. Both proteases are the main protease and the papain-like protease. It binds to the papain-like protease to less strongly than the protein, less extensively to papain-like protease than the main protease. Therefore, it might play a role in preventing the post-translational processing of viral proteins. It also binds well to the N protein, as well as to the non-structural protein 14, which is essential in transcription and replication of the viral genome and viral assembly. The second drug is remdesivir. Remdesivir binds well to the RNA-dependent RNA polymerase, being actually originally an adenosine analog. So this is expected. It also binds to the two cellular receptors, the human S2 and the TM BRIS2, which denote that it's possible to have a role in blocking cellular receptor necessary for cell entry. It also binds well to the proteases, both proteases, main protease and papain-like protease, as well as to the non-structural protein 14, which is suggesting of having a role in inhibiting different steps in viral replication. For the rest of the drugs, chloroquine and hydroxychloroquine showed considerably high binding affinities to main protease, M protein, ACE2, and TMPRSS2. However, their binding affinities were lower than those of ivermectin and remdesivir for all tested proteins. Lopinavir also docked well with membrane. However, upon simulation, it formed unstable complex. Finally, the favipiravir did not show any good binding affinity to any of the tested proteins, although it was reported to be of a good result with patients in some clinical studies. So finally, the conclusion of our study is that both ivermectin and remdesivir could be potential drugs for treatment of COVID-19, because both of them exhibit multidisciplinary inhibitory effect at both viral entry and the post-entry stages. So ivermectin binds to the viral S protein and the two cell receptors S2 and TMPRSS2, indicating it may be involved in inhibiting the viral entry. Also, it binds well to the main protease, babin-like protease, which means it may play a role in preventing post-translation processing of viral polyproteins. Additionally, ivermectin effectively binds to the viral N-phosphoprotein and non-structural protein 14. This is suggestive of its role in inhibiting viral replication and assembly. And finally, remdesivir may be involved in inhibiting post-entry mechanisms, being found to have high binding affinity to N protein, M protein, babin-like protease, main protease, RNA-dependent RNA polymerase, and non-structural protein. Thank you for listening to me. I'm accepting your questions, and more details are found in the publication which I showed in the first slide.